Welcome everybody to the 2021 Northeastern IPM Center Research Update Conference. I am Jana Hexter. I work at the Northeastern IPM Center, and I'm glad that you could be here today. This session is being recorded, and we have a wonderful lineup. So you can always come back and watch the session again if there's something that particularly piqued your interest. And I am having a problem moving my screen. There we go. Okay. So the webinar is being recorded and it is available at this link, but don't worry about keeping track of that link because I will send you an email. If you're registered for the update conference, I'll send you an email with the link. If you have questions, which I hope you do, and I'm sure you will, we ask that you use the Q&A feature. It helps us to keep track of things, or it helps me to keep track of things a little better. So if you click on your Zoom screen, the toolbar, in the middle, there's a feature that says Q&A. And if you click on that, you can ask a question. You can do so anonymously, so you don't have to feel shy about what questions come up for you. And if you want to make a comment or share a link with somebody else, please feel free to use the chat feature and make comments as we go along. We also have a Find a Colleague feature on our website. So if you go to this link, and I'll also send a copy of that to you, you can put a profile of yourself up on there. And it's a good way for us to increase collaboration, so that if there's a particular project that you are looking for a collaborator on, or you're interested in collaborating maybe with one of the presenters, many of them are here on this site. And the whole goal of this conference is to keep you up to date with what's happening in the Northeast, but also to create more collaboration between folks across the region. So with that, I will stop sharing my screen, and we will begin with the first presentation, which is by Mahfouz Rahman from West Virginia University, and it's Integrated Options for Managing Soil-Borne Diseases of Strawberry. And I have to apologize, for some reason this recording is a little bit, the sound on it is a little bit low, so you may want to turn up the sound on your computer so that you can hear it.
My name is Mahfouz Rahman. I'm a plant pathology extension specialist at West Virginia University. This project was taken to determine the efficacy of several biological and biorational treatments in managing soil-borne disease of strawberry, especially black root rot complex and crown rot. These diseases are extremely difficult to control without long-term rotation or fumigation with synthetic fumigants. However, many small growers and UP growers tend to grow strawberries in the same piece of land repeatedly, and regulations or need to hiring a custom applicators do not allow them using synthetic fumigants. As you know, strawberry fruit producers in most cases use flag plants bought from flag producers. We worked with a major flag supplier in the Mid-Atlantic and Northeast to produce flag plants in beneficial microbe-inoculated medium. So the beneficial microbe we used, the name is TerraGrow, which is a cocktail of 6 different beneficial microbes. So we used pasteurized medium and non-pasteurized medium to inoculate with TerraGrow. And also in flag production phase, in another batch of plants, we did not pasteurize and we did not inoculate that medium. So these flag plants are taken to the fruiting field. As you can see here, pasteurized medium and TerraGrow treated plants are taken to the non-treated fruiting field plot, and non-pasteurized medium that was treated with TerraGrow was taken to ASD, anaerobically disinfested soil plot, and non-treated plots. And the control plants are taken to either ASD or mustard cover crop or non-treated plots. So we had 6 different treatments, as I showed earlier, and those treatment nicknames are like first rise TerraGrow. This is to evaluate the higher colonization potential of TerraGrow. And the synergistic effect, regular, medium, treated with TerraGrow plus anaerobic soil disinfestation. And number 3 treatment was like ASD, anaerobic soil disinfestation, and then evaluation of regular colonization potential of TerraGrow, mustard cover crop, and non-treated check. So we planted these plants in the plot in 2018 fall, and then in the spring of 2019, we harvested strawberry fruit. As you can see here in the y-axis, we have fruit yield per plant expressed in gram, and in the x-axis, you see the different treatments with the nicknames. So we got only pasteurized medium and then a little bit of synergistic effect from regular medium and anaerobic soil disinfestation in the first year compared to other treatments. So these 2 treatments provided significantly higher yield. In the second year, you can see the overall fruit yield for plant was relatively low, but the difference among the treatments are wide than in the first year. So we got, from all the treatments, we got significantly higher fruit yield, except mustard cover crop compared to the non-treated check. One interesting observation from this trial was that you can see here in the anaerobic soil disinfested treatment, we got relatively low weed population that came through the planting holes. And here you can see we started with 20 plants in each plot, and at the end of the first year harvest, we saw many plants died in the non-treated plot, followed by mustard cover crop. In other treatments, we got relatively low number of plants died. So we kept those plants obviously for the second harvest, and then at the end of the second harvest, we saw more plants actually died in the non-treated plot, followed by the mustard cover crop, and other treatments also we had only very low number of plants died. So if you have any question, please send me an email. Thank you very much.
Wonderful. Thank you very much, Mahfouz. And we'll be having a Q&A session after the next video, which comes from presentation, which comes from John Nguatio, and he's from the University of Connecticut, exploring methods to enhance biocontrol of turfgrass diseases in the urban landscape.
Hello, my name is John Nguatio. I'm an associate professor of turfgrass pathology at the University of Connecticut. And I'm going to be speaking about some research I've been conducting with my graduate student, Kate Goodrich, and my collaborator, Dr. Joseph Roberts, whom you'll be hearing from later today. Today's topic is on the methods to enhance biocontrol of turfgrass diseases in the urban landscape. This is a topic which has become increasingly important in recent years, particularly in light of recent bans on the use of pesticides on turfgrass areas throughout the region. In Connecticut, we've had a ban on K-8 school grounds and daycare facilities since July 2010. And as you can imagine, this creates a challenge for turfgrass managers at these sites in dealing with various pests of these turfgrasses. Somewhat surprisingly, we have not seen an increase in the use of biological controls during this time. And this is likely due to the perception that biological controls are generally less efficacious and consistent compared to traditional pesticides. One of the challenges that we recognize in the efficacy of biological controls in turfgrass systems is the effect of competition between existing microbial communities on the leaf surfaces, such as bacteria, yeast, and fungi, with our applied biocontrol agents. These organisms are adapted to that particular turfgrass environment in the phylosphere, whereas our applied biocontrol agents are typically dormant bacterial cells or fungi that are applied and must break dormancy, initiate growth, reproduce, and build up their populations to have a beneficial effect. Of course, they have to do all of this amongst this pre-existing microbial community. This competition might oftentimes reduce the ability of biocontrol agents to actually establish and have a beneficial effect. So our research group has been looking at this and considering ways to overcome this challenge. And one concept we've been thinking about is the use of niche clearing, which we consider to be using products such as Xerotol 2.0. These are oxidizing agents, which can essentially eliminate bacterial cells or fungal cells on contact through an oxidation process. However, they have a very, very limited residual activity. Therefore, we could come in after an application of one of these products and apply our biocontrol agent, presumably under a lower competitive environment. So we've conducted a number of studies in the field in the greenhouse to test this hypothesis. And generally, those studies involve treatments where we either have niche clearing or we don't. And then those are followed up with an application of either biofungicides of Pseudomonas chlororaphis or Bacillus subtilis or a no biofungicide control. I'm going to focus here on our greenhouse results. And in the greenhouse, we actually quantified the amount of our biocontrol agent, Bacillus chlororaphis AFS 009. We quantified it on the leaf surfaces using a quantitative PCR assay with probes and primers specific for this particular organism. We had 4 applications of the treatments in this study, and you can see at 1 day after treatment of each of those, we have a peak corresponding with that application and a peak of the amount of the biocontrol agent at that time. Following that peak 1 day after treatment, each time we generally see a rapid reduction in the amount or the abundance of the biocontrol agent within 4 days after treatment, regardless of the application time. Interestingly, we do see a benefit to our niche clearing treatment. Our niche clearing treatment, shown here by the solid blue line, did actually provide an approximate 2-fold increase in the establishment of our biological control agent, Pseudomonas chlororaphis, at that 1 day after treatment. Okay, as we can see here at the 2nd and 3rd application timings. So we do see this initial increase in the establishment. However, those populations do still decline by 4 days after treatment. So we don't see a sustained benefit from the niche clearing effect. So to summarize our results of field and greenhouse work, in the field where we looked more at the efficacy of niche clearing on improving biocontrols against gray leaf spot of foliar disease of perennial ryegrass, we did not see improved efficacy associated with niche clearing treatments. However, in the greenhouse studies, we did see niche clearing did increase the initial establishment of Pseudomonas chlororaphis at 1 day after treatment. However, the effect was temporary in these treatments. The amount of these biocontrol agents did typically decrease by 4 days after treatment. Based on these results, we find that the niche clearing treatments look promising. However, we recognize we need to look at additional factors which may increase the persistence of these biocontrol agents beyond 4 days after treatment. So in future research, we hope to look at additional biocontrol agents which may be better suited to this environment on the phylosphere, as well as nitrogen and carbon nutritional inputs, which also might help sustain biocontrol agents for a longer period of time. With that, I'd like to thank you for your time and take any questions.
All right, wonderful. No, thank you. Actually, we don't have any open questions at the moment. Please feel free to put your questions into the Q&A. We have like little 5-minute breaks at the end of each half hour, and I'm happy to circle back around if John Mafuz is still here as if you put your questions in. So actually we're right on target to start the next presentation by Terence Bradshaw from the University of Vermont. And his presentation is New England Cider Apple Program.
Good morning. I'm Dr. Terrence Bradshaw from the University of Vermont. We're going to be discussing the Northeast Cider Apple Project, which is a Northeast SARE funded research and education grant running from 2019 through 2022. We began studying cider apple production a few years ago at the University of Vermont and at other universities around the region, mainly because we were seeing a real change in terms of a new market demand for fruit that weren't previously grown. In fact, for many years, much of our work, whether it be in IPM or horticultural production, was oriented toward avoiding the production of what were then low-value cider apples. Beginning around 2013 or 14, the market for cider, fermented cider, was rapidly increasing to the point where cideries were having a difficult time accessing the fruit they needed to keep supplies in stock. So we began doing some work at the University of Vermont, and as I mentioned elsewhere, to study some of the production strategies to help address that issue, price point. So we started NECAP, the Northeast Cider Apple Production Project, with a few different objectives. Number one, a number of the European type cider apple varieties or cultivars have a strong biennial tendency. Large flush of fruit one year, almost no fruit the next year. And that's very difficult to try to manage and maintain profitability if you're planting a high-cost, high-value orchard and you have cideries that are planning on annual production of fruit. So we have a few different projects going on in both Maine and Vermont, looking at a number of different strategies to try to bring those biennial trees into more of an annual bearing cycle, or at least to take the peaks and valleys and shorten them up a little bit. So we're looking at some plant growth regulators, or we have looked at some plant growth regulators to try to influence fruit bud production. Looking at mechanical hedging, which changes the hormone flow within the tree and kind of brute forces the tree into producing more fruit buds. And up in Maine, they're looking at string thinning, where flowers are removed at bloom prior to becoming fruitlets. That prevents the tree from oversetting fruit, overstressing itself, and therefore not producing fruit for the following year. We're also evaluating some of the unique pest incidents and disease susceptibility of cider apple cultivars. And the eventual goal is to see whether or not we need to make adjustments to some of the models that we use for managing diseases in our orchards, and whether or not we can try to adjust our spray programs and reduce the application of pesticides, because we don't have the need for cosmetically perfect fruit. We're also making some systematic cider cultivar observations, and this is done really region-wide by collecting input from growers, as opposed to having a single research orchard where we can set up, replicates of certain number of n equals 5 of 40 different varieties or whatever. We don't really have that orchard in New England, that ideal orchard, and B, there are many growers who are already growing these fruit and are generating data and observations. And so we are working with those growers to pull together some of their data, some of their observations. And at the end of this project, we'll be presenting a comprehensive list of the different cultivars that are grown in the region and some of the growth characteristics and management of them. And the real goal at the end of this is to build a stronger community of cider orchards and cideries to help benefit both industries. There's a number of outputs that we will be putting out throughout the time of this project. Of course, there's the Northeast SARE project overview and project report page. We are together as a team, and John Clements from UMass is working as our extension person to help post much of our material on the e-extension apples page, and there's a specific cider apple category. And my colleague Liz Garofalo at UMass also has a YouTube channel where she highlights a number of the observations that she sees throughout the season, and that's been a good kind of public relations outreach piece. So with that, we'll wrap up and take questions.
Wonderful. Thank you very much, Terrence. And our next presenter is Kim Skirm, and she's talking about a Varroa Mite IPM program for New England honey beekeepers. And he's joining us from the Massachusetts Department of Ag Resources. And if you have questions for Terrence or for Kim, please put them in the Q&A. And I see there's been a couple of questions come in from Afuz and John. We may circle back around to those. So this is Kim's presentation.
Hi, everyone. My name is Kim Skirm. I'm the Massachusetts State Apiarist, and I'm joined by
Jen Lund, the Maine State Apiarist, and we're here today to present our grant, a Varroa Mite IPM program for New England beekeepers. Varroa mites are originally from Southeast Asia. They were first detected in the US in the late 1980s, and they jumped from another species of honeybee onto the one that we manage, the Western honeybee. And these mites feed on the fat bodies of bees, which are very important. The fat bodies are very important for detoxification of toxic substances, immune response, as well as a backup energy supply. And they're one of the largest ectoparasite to host relationships. They're very large compared to their host. When we ask beekeepers what their biggest challenge is, they always answer the top choice is always Varroa mites. That's both in Maine, Massachusetts, and actually around the country. And so the cornerstone to any IPM program, of course, is monitoring, knowing where your pest levels are, and that can dictate when you need to perform treatments and what you need to do to control them. So we do find that beekeepers who are not monitoring have higher losses on average than those that are monitoring. And not all monitoring is the same. So here you can see other methods of monitoring compared to alcohol wash. And alcohol wash is by far, beekeepers who are doing that have by far fewer losses than those that are using other methods. And then we also know timing those treatments or pairing those treatments with what you're finding in monitoring is also equally important. So those that are not treating for Varroa mites at all have much higher losses, up around 75, 76%, while those that are using both prevention and intervention methods, so those are preventing mites from increasing in the hive, and also intervening when your mite levels get high enough to require a miticide treatment, have those people who are combining those two methods have lower losses on average than those that are not treating or just using either prevention or intervention.
Now, it's very important whenever you're managing any sort of pest insect, right, to have a really great sampling tool. And early on, we noticed that beekeepers lacked accessibility and ability to find this tool. So what we did was we launched this Varroa Mite monitoring jar that you see here, very simple design, as a way for them to be able to sample their hives to determine the threshold, the level within the hives of the mite populations. We paired this sampling device with this informational brochure that you see on the right. And this brochure is really specific to go through all the different biology, kind of key biology pieces of information that beekeepers need to know, mixed in with a very visual tutorial of how to do an alcohol wash, how to use that sampling tool, that jar set up to monitor for Varroa mite populations. And then inside of the brochure, we're really excited because, again, we paired this information of sampling with the treatment options that are available. And this was a collaboration between ourselves, our institutions, and UMass Amherst Extension to be able to produce this very visual tool for beekeepers that has a ton of information here listed for them. It's kind of a one-stop shop go-to for Varroa Mite IPM. In addition to this tool, we went a step further and offered in-person workshops and trainings, like you see with Jen here, showing beekeepers in the field how to actually do mite wash samples, as well as showing them just how large these Varroa mites are in comparison to a bee. And then with our workshops, if you'll notice behind that photo, this is an example of an IPM plan that we would encourage beekeepers to create for their own individual apiaries. So that way they can walk away with not only the sampling device, hands-on learning options with training on how to do a sample, take a Varroa mite sample in their hives, and then of course a plan for what to do if those thresholds were met with the mite population. And what we see overall since we've had this grant, not only the impact during the grant, but it has been sustained throughout that long after, And you can see a really great metrics here for both in-person education as well as online education. And then we're actually still producing these brochures and still handing out these Varroa Mite jars even to date, because it's been such a popular program in our individual states. And even our colleagues in other states in New England in the Northeast have started to take on this program. And you'll notice at the bottom here, we did partner in this grant with the Mite Check Map, which is an online map platform that allows beekeepers to take the data they've collected during their own sampling of Varroa mites and input that into a map so they can see other relationships with beekeepers in their area. In the future after COVID, we'd love to be able to continue these in-person workshops. And we're noticing a trend with this, both in what you see here in the blue, which are people that monitor for Varroa mites. As you can see, that has increased since the start of this grant project, and we expect it to continue to increase. We hope it does. And also with those that treated, which has been maintained fairly stable throughout. Now we can start really bridging the gap between these two aspects, right? Because we don't want to just have folks monitoring and not being able to do treatments as they need them. And of course, not doing treatments and not having the information of the monitoring to know if the treatments were even warranted. So this is a really great trend to be able to see in our data. And then finally, I'll leave you with the website for our project if you would like to learn more about the work that we've done, the impact of this work. And then, of course, you'll see we've got a series of webinars here. There's links to the tools that we discussed. And overall, just really excited to be able to share this with you today, and hope that if you have future questions or if you need any more information, you won't hesitate to reach out to us. Thank you so much for your time, and I hope you have a great rest of your week.
Wonderful. Thank you very much. And I saw there was a couple of questions that that came in the Q&A, and our speakers have actually answered them in typing, but actually one just came in for Jen and Kim. Do you find beekeeper resistance to the method because it kills bees?
This is a great question. Yes, we have had definitely some beekeepers be a little bit nervous about using the alcohol wash because it does kill bees. We are seeing really interesting sort of a trend now of people understanding that if you don't do some sort of monitoring sample, that the end result could be that you lose all the bees in the hive. So I think we're really starting to bridge the gap now of the maybe the sacrificial lambs of these bees in the sample being kind of that sign of whether you need to treat or if you have a problem in the hive. But yeah, initially this was a little hard, I think, for people to sort of take on, and nationwide doing a sample is part of beekeeping, but typically they've been using powdered sugar as the media instead of alcohol, which obviously doesn't kill bees as much as powdered sugar wouldn't kill them as much as the alcohol. But I think we're finally kind of bridging that and maybe crossing that threshold where this may be okay. So it's really been exciting to see this change. But yeah.
Wonderful. Great. Thank you. And for those of you who are interested in this topic and did not see the series of webinars that Kim and Jen put together, they were wonderful. They're fun, they're amusing, and chock full of information. So they were a definite hit on our webinar series. So if you haven't had a chance and you're interested, circle back around and go watch those. And they're also extremely helpful and open to people contacting them with questions and comments. So great. It's good to see you and to see the bee background.
Yes, I know. You too. I know. I feel like we're getting so close to this in spring. I keep looking out every day. I'm like, oh my gosh, is there more green coming up out there? We so need it desperately. So yeah, so good to see you.
Yeah. And I heard the other day, I don't know if this is correct, but I heard the other day that 1 in 3 bites of food that we put in mouth, or a third of the food that we put in our mouth, comes from pollinators. And that really, I don't know if that's actually accurate, but it stayed with me when I heard it. So
yeah, no, yeah.
So okay, all right. So we're going to move on, and the next presentation is by Sam Anderson, and his topic is two-spotted spider mite in New York City urban agriculture, and he comes to us from the Cornell Cooperative Extension.
Hello, my name is Sam Anderson. I'm an urban agriculture specialist with Cornell Cooperative Extension based in New York City. And a lot of the work I do is IPM work with urban farms, or 30 or 40 urban farms, sort of commercial scale or market scale urban farms spread across the 5 boroughs. And most of them grow vegetables and most of them grow tomatoes. And the top pest by far of tomatoes in New York City is two-spotted spider mite. And that's not just in greenhouses where we might expect to find it, and not just in high tunnels, but also in, our equivalent of field tomatoes, just outdoor tomatoes. And this is true all over the city. It's true on rooftop farms. It's true at ground level farms in every kind of soil, on the little islands. It's true at almost every farm. In fact, 75% of sites that I scouted in 2019 with tomatoes had significant two-spotted spider mite damage. Most of the other ones that didn't have bad damage still had a presence of two-spotted spider mite. So it's all over the city. And I think that's, given that I only have 5 minutes here, that's the most interesting thing, is just the fact that two-spotted spider mite is so present here, and present in damaging levels on tomatoes, cucumbers, eggplant, and beans, which aren't commercially important to anybody here, but are important because I'm going to tell you why in a minute. So that's the big question. Why is it so damaging on urban farms in New York City, on crops where it wouldn't be damaging most of the time outside the city in this region, except in greenhouses or high tunnels? So it could be the urban heat island effect, which makes, it doesn't necessarily make like heat waves hotter, although there are probably microclimates where it does, but it does make nighttime lows not as low, 5 to 7 degrees Fahrenheit warmer, so stuff doesn't get killed off as easily over the winter. That probably explains a number of the other interesting pests that we have, like harlequin bug. But seems like there's more going on for two-spotted spider mite. And one of the questions is, is it a lack of natural enemies? And if so, which natural enemies are missing here? We've got lots of ladybugs and lacewings, and then we actually have lots of aureus, a minute pirate bug. But what else what might be missing? Just to show more examples of how common it is. It's on cucumbers a lot as well, in high tunnels where you might expect it to be an issue, but also in the field. On eggplants, that one on the left is particularly hard hit, and also on house plants. So somebody sent me a picture of their lemon tree with bad two-spotted spider mite damage on the right there. So I see it on street plantings, on trees. Once you know what the damage looks like, and once you start knowing how to look for the mites themselves, and once you happen to carry a hand lens with you everywhere you go, extension life, you start finding them all over the place. And so I put together a little partnership grant to do a scouting protocol to put bush beans at row ends of tomatoes on a few farms. It's much easier to scout bush beans than tomatoes. Damage, you're going to see it sooner. And so then the idea was to respond to that by releasing biocontrols. We did Folaces, Feltiella kerasuga, which is a predatory midge, and Persimilis. And then also some habitat plantings, which we didn't really get into last year, but hopefully we'll get to do some more this year, and trying to see what natural enemies that are already present that we might be able to attract and maintain. And so just the last little interesting point on that. So the stuff we released, we just found out we need to release it before we see any damage at all. The Feltiella, or the Folaces and the Feltiella, have to be released very early as a preventative measure. We were a couple weeks late on it. And the Persimilis, the moment that we see damage, we need to be ordering Persimilis. I think they just weren't able to keep up with the spider mites at any of the sites probably. But we did have this interesting case at this one farm, Red Hook Farms here, where they planted bush beans the whole length of one of their tomato rows in this system that you see here. They came up with their own sort of single strand lower and lean system, and planted bush beans the whole length of it. And those tomatoes were the least affected by far by two-spotted spider mite of all the places that I scouted this year. And interestingly, on those on the beans next to the tomatoes, and on the tomatoes themselves, early in the year, there were lots of aureus of minute pirate bug, both adult and larvae. And so that, is sort of an open question. It's just one farm, just one year, but something to watch for in the future is maybe attracting aureus through a sort of something that functions like a banker plant, which might be how the beans functioned in that case, or maybe the beans were flowering early and they retracted the pollen, but it seems more like it was sort of a banker plant. But finding ways to attract aureus, that might be a key natural enemy that helps to keep spider mite in check without having to order and Persimmons or other natural enemies. And then last question, just can we help some of them to overwinter in significant numbers, especially Foliaceae? Persimmons won't overwinter, but Foliaceae might. So that's an open question. I've run over my time, sorry, despite speaking really quickly. My information's at the bottom. I'd love to hear thoughts anybody has, other stuff we might want to try, or other reasons why, any theories anybody has about why spider mite might be an issue in the city. Always love to hear it. Thank you very much.
Great. And we actually have a couple of questions that have come in, and we have a time scheduled for that in 5 minutes once we have listened to Joseph Roberts' presentation on exploring methods to enhance biocontrol of turfgrass diseases in the urban landscape. And he's from the University of Maryland.
Hi, my name is Joe Roberts, and I'm the turfgrass pathologist at Clemson University. Today I'm going to talk about some work that I participated in with Joe Doherty while at the University of Maryland, collaborating with researchers from the University of Connecticut, John Guigiato and Caitlin Goodridge. This stemmed from growing concerns of pesticide use, particularly on turf grasses used in the urban landscape. And there has been legislation enacted over the recent years in some states, as well as certain local municipalities, banning the use of synthetic pesticides on turf grasses used in public landscapes, schools, municipal sports fields, as well as home lawns. And seeing these bans, we do see that there is a need for more research on understanding organic means of pest control in turfgrass systems. Now, for our project, we chose to work on brown patch, one of the most common diseases of cool season turfgrass is used in the landscape. And the causal agent of this particular disease is Rhizoctonia solani. And we do have commercially available biological control measures that are available with limited efficacy data. So we do think that if we were to develop additional research, we could hopefully yield more reliable efficacy data and thus, adopt or higher adoption rates of these compounds in the future. Now, for our project, we decided to look at a surface sterilization or a surface disinfestation to try to knock down the existing bacterial and fungal population on the turfgrass leaf prior to treating with a biocontrol agent. And the thought is that if we were able to knock down that population for a certain period of time before that rebound, we could come in with our biological control and thus have a greater niche for that introduced biological and enhanced establishment that thus may translate to a higher efficacy against the brown patch pathogen. Now, we did a combination of both greenhouse and field-based studies, and what I'm highlighting here is some work that was done in the greenhouse. This is just one run of our greenhouse experiment looking at brown patch control measures for perennial ryegrass. And again, what we're looking at is a combination of treatments, looking at xylitol, the active ingredient being hydrogen peroxide, followed by a Rhapsody or Bacillus QST 713 strain known to impact brown patch disease. And then we also have xylitol by none. Then we have a none followed by Rhapsody alone. And then we have a none by none combination. Now, to set up this figure, what we're looking at here is actually the log 10 base recovery of the QST 713 DNA per gram of turf tissue. And what we developed this utilizing or by adapting a protocol previously developed by Mendes et al. In 2018 with a qPCR method that has been successful, as you can see here, in documenting recovery of that introduced biological antagonist. So from the get-go, you can see that both of our Rhapsody treatments have an increase in recovery following that first application. Within a few days, we do see a decline in recovery to the point here just prior to 4 November, we actually see an increase again. That's actually following a second application before it declines again following that second application. What you can also see here is that while our initial hypothesis of actually utilizing xylitol to increase establishment and efficacy, we're actually seeing the opposite with xylitol actually lowering recovery of that QST 713 DNA recovery. So indicating that we're likely lowering that population of that Rhapsody introduced antagonist to the point where we need to look at these combinations even further. Now, I did say that we apply that xylitol 3 hours prior to the Rhapsody application, and we may be looking at some additional timings in the future to see if there is an impact of that timing. But we also have looked at these compounds in the field against brown patch disease. And here we're showing some data from 2020, looking at brown patch severity as a percent per plot area. And what we have is 4 different rating dates across the season, with our highest amount of brown patch observed on the 3rd of August, with a little over 50% in the non-treated check. And what I want to highlight here is that while our synthetic heritage or azoxystrobin compound utilized as our positive control showed very good efficacy across all dates, we did see some enhanced control for Rhapsody, or that same biological antagonist, applied on a 7-day interval on 2 of the dates where we had moderate pressure. Now, this is with no xeritol pretreatment. We hope to adapt some work in the future looking at these in addition to various intervals to enhance efficacy. Now, we want to look at additional cultural practices in combination with biological controls and hopefully adapt this to additional turfgrass diseases in the future.
Wonderful, thank you very much. And we had a couple of questions come in for Sam. Susanna has asked, what is the value or volume of crops produced on urban rooftops or similar farms? And are there commercial rooftop farm operations in New York City? And Deb Grantham actually asked a similar kind of related question, which is what is included in urban ag? Is that rooftop, for example?
Yes, I was just starting to type it, but it'd be easier to answer it this way. So I guess, so firstly, what's included in urban ag, it can be everything from, I mean, community gardens, which we have over 700 in New York City, are a big part of it, but most of what we work with, my colleague Yolanda and I, are commercial urban ag. And for our purposes, we loosely define that as anyone growing $1,000 or more of, growing and distributing $1,000 or more of agricultural product. Not that we're really counting most of the time. And most of those, a lot of them are nonprofits. In fact, well over half are nonprofits, but the money goes back into their budget and goes to pay for staff and things the same way it would for a for-profit farm. And we do have, to talk about rooftop, for example, we have a number of rooftop farms in the city. Some of them are very visible, well, not visible from the ground, but visible in terms of people know about them and they're visited by a lot of people. But the majority of the urban farms are ground level in a sort of a constructed soil. So something that is brought onto the site. What's underneath might be a parking lot. It might be just sort of a low quality soil, or they're concerned about lead contamination. So soil of some sort is brought in. And
what were trap catches yesterday?
Sorry, let me see if I can make that stop.
Yeah.
That's okay, carry on.
And so, I thought it was going to be a soils question. And I was like, I don't think I have time to, I can talk a long time about the unique urban ag soils. But anyways, the point is there are a lot of different ways that urban agriculture looks in the city. So there are some rooftop farms, but the majority of farms right now are more ground level. And there's some hydroponic and indoor farms as well, but still, again, well over half are ground level soil-based diversified vegetable farms, basically. Yeah.
And I have one little, I know we only have a minute till we have to move on, but I have one more quick question is, are they evenly distributed, or are they more concentrated in some boroughs than others in the city? Like, do you have more in Staten Island or the Bronx, or I presume not so many in Manhattan? What's the distribution? Yeah,
so you're right, not so many in Manhattan. A lot of community gardens in Manhattan, but they just aren't at the scale. Whereas basically, like, I can't spend half an hour necessarily talking to somebody about their 2 tomato plants, but I can talk about their 100 tomato plants. And so that scale, bunch of them in Brooklyn and the Bronx, those are probably where most of them are. Not as much on Staten Island as you might think, but there are 4 farms on Staten Island we work with, and a few in Queens, but really Brooklyn and the Bronx are the hot spots. But yeah, they're all over, and the smaller islands have them. I mean, they're scattered all over the city, and usually not where you'd expect.
Great, lovely, thank you. And it's lovely to hear the baby gurgling in the background.
Can you
so.
hear? Yeah.
Added bonus for signing in today, you get to hear a baby gurgling in the background.
Yes, it does.
Lovely, thanks.
My colleagues are familiar. Yeah.
All right, great. And our next presentation is by Daphne Monroe. And she is talking about the application of shell hash cover as a deterrent of cow nose ray predation on hard clam farms. And she's with Rutgers University, and this is a project that was funded through Northeast SARE.
Hello, everyone. My name is Daphne Monroe. I'm an associate professor at Rutgers University, and I'm happy to be here today to share some information about a project my colleagues and I are working on with our farm partner, Mr. Dale Parsons of Parsons Seafood. Dale came to us just over a year ago with the idea behind this project, and we're grateful to him and for his continued partnership, and to the Northeast SARE program for supporting this effort. First, some background about our motivation. This photo shows a typical clam farm in New Jersey. These farmers are working the farm at low tide, placing net screens over their clam crop to protect it from predators. This method is highly effective on farm leases like this one that can be worked at low tide. Along the Atlantic coast of New Jersey, shown in the map on the left, there are about 2,500 acres of leased bottom for growing shellfish. About half of that is in water shallow enough that it can be farmed using the methods discussed in the previous slide. The remaining lease areas are too deep to be farmed that way. Traditionally, those deeper leases were farmed by planting clam seed unprotected on these leases and returning to harvest with tongs some years later. That method no longer works because of a novel predator that has become a persistent problem for clam farmers in New Jersey, and consequently about half of the leased bottom that's available to our farmers is no longer in use. The predator culprit that I'm talking about is the cow nose ray. Seen on the left photo, that ray is picking up a full-size oyster in its jaws. These predators can grab shellfish, including clams, right out of the bottom and chew them up using hard, crushing mouth plates. Historically, this species was not an issue for clam farmers in New Jersey, but as climate warms, large schools have been seen migrating further north and are now a big issue for our clam farmers, and one that prevents them from using those deeper lease areas in New Jersey that I was just talking about. The solution that Dale brought to us and that we are testing in this project is planting shell on the bottom. Our hypothesis is that planted shell will protect small clam seed and deter cow nose ray predation, because it will make it hard for the cow nose ray to flap their wings, loosen sediment, and get at the clams in the bottom. It would also be able to be used on those deep water leases because the farmers could still use tongs to harvest the crop when it's ready to be harvested. The added bonus is that we know from other research that shell hash improves the habitat for clam growth. So it's a double win. In this photo, you can see my colleague Mott's, Tom Grotus, on the left, and Dale Parsons, our farm partner, carrying a clam net screen to set up one of our experimental plots. You can also see on the right, if you look closely, a mound of planted shell poking out of the water at extreme low tide. We set up replicate control, netted and shelled plots, then planted them with clam seed last fall. We will return to our plots this coming spring to observe cow nose ray behavior at these sites. The photo above is not our team. It's a crew from VIMS tagging a cow nose ray. We'll take a similar approach and tag rays when they migrate in to New Jersey this spring, and set up a listening array around our experimental plots that will allow us to triangulate their location and behavior as they interact with our three treatment types. In addition to watching ray behavior at our plots, we will sample the clams to evaluate differences in growth and survival among our treatments. Cow nose rays crush clams in such a way that the remaining dead shells can also be used to estimate ray predation rates. And we will be collecting dead shells at our sites to also use those as a tool to evaluate predation. thank you for joining today. I hope to present some exciting results in the near future. Again, I'm grateful for my colleagues Lisa Calvo, Tom Grotus, Jason Morrison from Rutgers, and especially to Dale Parsons, our farm partner, and of course the Northeast SARE program for supporting this work.
Thank you, Daphne. That was really interesting. So our next presentation is by Lisa Tewksbury, and it's an update on biological control of swallow warts. She's with the University of Rhode Island, and this was funded through an EIPM grant.
Hello, I'm going to give you an update on the biological control of invasive swallowworts project from the University of Rhode Island's Biocontrol Lab. I'm Lisa Tewksbury, and this is a project we've been working on for quite a few years. Invasive swallowworts are composed of two different species, black and pale swallowwort. And these pictures show that it can be quite a problem in pastures, and it's also a problem in natural areas, roadside edges, field edges. As I said, invasive swallowworts, there's two species. Black swallowwort is Vincetoxicum nigrum, and pale swallowwort, Vincetoxicum rossicum. You can see nigrum, the black swallowwort, has a darker purple flower, and pale swallowwort is pink. They're members of the milkweed family. They have windblown seeds, perennial herbaceous plants, a vining growth habit, and monoculture shade out native plants and threaten habitat of endangered species. They're also toxic to livestock. And one of our graduate students found that as well as competing with milkweeds in pastures, the females are also confused by the swallowwort and lay some of their eggs on swallowwort, which unfortunately doesn't allow complete development of the monarch larvae. So the biocontrol agent that we worked with was Hypena opulenta, and the host plant was previously unknown before Aaron Weed's graduate program allowed him to collect this insect in 2006 in Ukraine. It has multiple generations a year and a 5 to 6 week life cycle at 20 degrees C. The host range testing that we did showed that the hosts are restricted to the genus Vincetoxicum. And after going through the tag process, which is a technical advisory group, and then the USDA needs to go through the process of getting a permit. So after the technical advisory group approved, the first releases were made in Canada in 2014. And then after the USDA gave us a permit, the first releases in the US were in 2017. And we made releases in Rhode Island and Massachusetts first. And this was the 2017 Rhode Island release in Charlestown, Rhode Island, in a conservation property. And the release was in the fall. And we did obtain one moth that emerged on June 1st, which gave us an indication of the timing in Rhode Island. And then in 2018, we made additional releases of larvae, and we had set them up, set the cages up in full sun, partial sun, and shade. And this graph shows you that the higher number of damaged leaves was found in the shady spots, the black line there, and the full sun, which is the red line, we had much less feeding damage, so less larval survival. Because 2018 was such a hot summer, we did have high mortality, and we decided to go the way that Canadian releases had begun going, which is to release adults. So in 2019, we released adults a little earlier in the year, and you can see the feeding damage on the right, and we removed the cages after approximately 3 weeks. In the Charlestown site, we did get some emergence in 2020, and we made additional releases in 2020. And this gives, again, at the Charlestown site, you can see that the daily minimum temperatures are in the gray line, and that orange line going across shows the lower developmental threshold for Hypena larvae. So in Charlestown, it's a colder area. During that time from June through July, there were many times when the temperature was below the developmental threshold. Anyway, after about 3 weeks, we had about 70% damage at that site. So the lessons we've learned are that Hypena opulenta does much better in the shade. The adults will be flying in June. Adult releases were much more successful than larval releases. And the earlier that we can make the releases will improve our chances of getting a second generation. Research from Canada on photoperiod indicates we may only have one generation in our area. And we did, in the course of this research, also discover a plant pathogen, which we're working with Matt Tankos from the USDA-ARS to look at, and Lisa Castleberry identified it as Passiflora balanicki, and others have also isolated a Calotatricum from these leaves. And thank you very much. I appreciate your attention.
Well, thank you very much. They're both very different and interesting presentations. So we have one question for Daphne. Deb asked is the cow-nose ray native, and how has the habitat of native species versus aquatic farming balanced, or how is it balanced? She says she's not trying to be confrontational. She's just curious about how those two things are balanced.
Yeah, that's a good question. The cow-nose ray is in fact native, and it's not that it's never been in New Jersey, but a couple of things are happening with this species. Is the population numbers are increasing as hunting pressure from large sharks is increasing, because there's been moratoriums on large sharks for a long time. And so there's been a population increase, but there's also been global warming, which is allowing these cow-nose rays to move further north. They go as far north as Massachusetts now. And so it is a native species, but its range is expanding. And so that's where the problem is coming in here for the farmers in New Jersey. As far as native and non-native in the shellfish aquaculture world, they're pretty aware, they're pretty conscious of invasive species, and the farmers here are not allowed to grow anything other than native shellfish species. I think that's what the question is. Is that right, Janet?
Yeah, I think it's the question of how do you balance the needs of a native fish that's moving north versus the ones that you want to farm here. So that's
sure. Yeah. I mean, so I think that's kind of what we're trying to get at here is because in some other places the farmers have looked at culling the predator, so hunting the cow-nose ray or fishing for it, increasing the fishing effort. And instead of going that direction, we're just trying to find a way to deter the predation. So the cow-nose rays can still come here, but they can't get at the farmed clams. They might be able to get at wild clams and other, other invertebrates that they're after, but we're just trying to keep them off the farm plots. That's all. Yeah. And we're not trying to do it in a way that harms the rays themselves. We're cognizant of that. Yeah.
Really interesting. So that's the first IPM project I've seen that's based in the oceans, and so it's interesting for me. So
yeah, this has been a lot of fun to hear about all these other terrestrial farmers.
Yeah.
I'm the black sheep today, obviously.
Great,
thanks.
All right, thank you. All right, so we should move on because it's 11:29. Our next presentation is by Veronica Yurchak. And she's with the University of Maryland, and her project is on creating an eco-friendly pest suppression program in sweet corn, and it's a project funded by Northeastern.
Hi everyone, I'm Veronica Yurchak. I'm a graduate student at the University of Maryland, and my project is titled Creating an Eco-Friendly Pest Suppression Program in Sweet Corn. So the goals of this project are to effectively suppress weeds using a perennial living mulch and cover crop residues, to eliminate strip tillage using winter-killed cover crop species, and to reduce competition with the living mulch to avoid any yield loss, which is often a problem when incorporating living mulch into vegetable production. And then also to provide a flowering resource to attract predators and parasitoids in an attempt to reduce pest pressure and insect damage to sweet corn ears. And so in this study, I have 4 treatments, 2 living mulch treatments, 1 which combines alternating sections of red clover living mulch with forage radish. And the idea in this treatment is that the forage radish will naturally die in the winter, resulting in bare ground planting rows by spring without the use of strip tillage, and then a red clover living mulch growing between our sweet corn rows. The same idea applies for the living mulch rye treatment. So we have alternating sections of red clover living mulch with rye, and this rye is terminated in the spring via roller crimping. And so then the sweet corn is planted into the strips of rye surface residue. In the no-till treatment, a mixture of rye and crimson clover is planted in the fall and terminated via roller crimping in the spring, and then that sweet corn is planted into that dead weed suppressive cover crop residue. And all of these treatments are compared to a conventionally tilled bare ground control treatment. And each treatment is further subdivided into 2 subplots. And so half of each plot receives an at-planting application of a residual herbicide, and the other half remains untreated. And this allows us to compare the weed suppression provided by these different cover crops and living mulches with and without a residual herbicide. And so just to give you a clearer picture of what these living mulch plots look like, this is a radish treatment, but as I said, the layout is the same for the radish and rye treatments. And so this is what this looks like in the fall after these cover crops are established. So we have 3 rows of forage radish alternating with 2 rows of red clover. And this pattern continues throughout the entire plot. And again, the idea here is that the radish will winter kill, and we can plant that sweet corn into the center of these bare ground strips. And all of our cover crops are planted at 6-inch spacing, such that we end up with 30-inch sweet corn rows with red clover growing between those rows. So looking at the weed suppression provided by our different treatments, here we have weed biomass at harvest, so about mid-August of last summer. You can see the conventional till treatment without herbicide had significantly more weeds than any of our other treatments. But what I really want to point out is that our 2 living mulch treatments with and without herbicide suppressed weeds as well as the conventional till with herbicide. So the living mulch really did an excellent job of suppressing weeds and did not require a residual herbicide to obtain adequate weed suppression all the way through the sweet corn growing season. By performing visual counts of insects weekly, we found that the most common predators in our system were minute pirate bugs, lady beetles, and wolf spiders, and the most common pests were flea beetles, tarnished plant bugs, and stink bugs, but we didn't see a significant difference in insects between our different treatments. Rating insect damage at harvest, we did find significantly more earworm damage in our no-till treatment. And I should mention, as far as yield, we didn't see any significant differences between any of our treatments. So some early conclusions are that the red clover living mulch provides equal weed suppression to tillage and herbicides, and competition with the living mulch did not result in reduced sweet corn yield. The presence of living mulch or cover crop residue didn't seem to impact the insect community, but we did find greater ear damage in our no-till treatment. And thank you, and I'd be happy to take any questions later on.
Lovely. Thank you very much, Veronica. So our next presentation is by Changlu Wang from Rutgers University, and he's presenting on monitoring and controlling house mouse infestations in apartment buildings. And it was work that was funded by the Northeast IPM Center. Here we go.
Hello, my name is Changlu Wang. Today I'm going to talk about assessing and controlling house mouse infestations in multi-family dwellings. This was an IPM issues project. The project period was between 2019 and 2020. As you may know, there are 3 common pests in homes. They are the German cockroach, bedbugs, and house mouse. In a recent inspection of more than 1,700 apartments in New Jersey, we found that the infestation rates of cockroaches, mice, and bedbugs were 37%, 16%, and 9% respectively. Mice is considered the most important public health pest in that they carry secondary pests and pathogens. They produce allergens, and they can cause significant economic damage. The prevalence of house mice in apartment buildings is partially because the lack of effective control methods and tools used by residents and by the property managers. As a result, chronic infestations often occur. The objectives of this study were to assess the spatial distribution of house mouse infestation in multi-family dwellings and to implement and evaluate building-wide house mouse preventative and control strategies for multi-unit dwellings. The study was conducted in 2 high-rise apartment buildings in New Jersey. We conducted building-wide resident interviews and house mouse monitoring to detect mouse activities. In each apartment, we placed 2 stations containing 2 different kinds of baits. And we also placed 3 dollops of chocolate spread to detect rodent activities. After rodents were found, we replaced the blank bait with rodenticide baits. Based on the monitoring results, we found that among those apartments with mouse activity, only 27% of the residents noticed the presence of mice. When comparing the 3 different blank baits, Detex is the least effective for detecting mice. Chocolate spread was the most effective bait for detecting mouse activities. We implemented the building-wide house mouse treatment for 2 months, then monitored the results for a 12-month period. The 2 buildings had very different results. In Trenton, the percent of apartments with mice activities increased after 12 months, whereas in Linden, the infestation rate decreased from 27.2% to 1.7%. The different results reflected the different maintenance levels of these 2 buildings. Linden building was well maintained, and there were very few defects in the structure of the building, so that mice couldn't invade the building easily. Whereas in Trenton, mice continued to invade the building during the study period. Interestingly, we found that most of the house mouse infestations were located among the first 3 floors. This diagram shows the spatial distribution of house mouse infestation in Trenton building. The orange and red bars are the ones with mouse activities. You can see that most of them are located at the lower 3 levels. In conclusion, we found that the mouse activities most likely occur in apartments on the lower 3 floors compared to those on floors above the first 3 floors. The chocolate spread is much more sensitive than the commercial rodent baits for detecting mouse activities. There are significant differences in the palatability of commercial rodent baits. Besides baiting, exclusion is also very important for mice management. Results of this study was recently published in Animals Journal. If you want more details, please read this paper. I'd like to thank Northeastern IPM Center for sponsoring this research, and thank you for your attention. If you have any questions, please write me an email at changluw@rutgers.edu.
Wonderful, thank you very much. And we actually have a couple of questions. One clarifying question for you, Changlu, is do you mean that residents didn't notice the mice presence at all, or they didn't notice it around the traps?
Yes, so most of the residents didn't notice mouse activity simply because, mice only are active during the night. So unless they caused already significant damage, otherwise, actually, many people didn't know. So that's something very interesting. People didn't like mice, but most people didn't know they have mice. Mm-hmm. So we need to do proactive monitor. Same for other pests.
Okay, great, thank you.
Sure.
And there's also a question from Afuz, from our first presenter actually, is does growing strawberries using aquaculture relieve some of the disease and pest issues?
Can you please say that again, Jana?
Yes, so does growing strawberries using aquaculture, and Deb said she wasn't sure of the terminology, relieve some of the disease and pest issues?
It does, you know, like if we grow, especially in an environment other than, you know, open field, that does control some of the diseases by itself due to the unfavorable environment. So yes, you know, like even inside the high tunnel and greenhouses, some of the disease severity is always lower than the open field.
Okay, great. I mean, thank you very much. All right, so we actually have time for one more question, and I believe there was one more for you from Afuz, actually. Oh, and I think you've answered it yeah, I think you actually answered it in the chat
That was from Deb, right?
Yeah. So great. All right, well, I think then we'll maybe move on, and it'll give us some more time at the end. So our next presentation is by Amara Dunn. She is with the New York State IPM program, and she's presenting on progress and updating the website Biological Control, a guide to natural enemies in North America. And we'll watch her presentation now. There we go.
Hello, my name is Amara Dunn, and I will be talking today about my progress updating the website Biological Control, a guide to natural enemies in North America. This is the homepage for the current Biological Control website, which was created by Dr. Tony Shelton, who's with the entomology department at Cornell University and just retired last fall. Before his retirement, he made arrangements for the New York State IPM program to take over this website so it can continue to be updated. So we have two goals as we're going to be migrating this website to the New York State IPM website. First, we want to improve the usefulness and usability of the site. And then second, we want to create and add new content. I wanted to make sure that we got feedback from stakeholders before we started making these changes. And so to do that, over the last year, I conducted 9 focus groups that included a total of 32 stakeholders representing 8 states plus the District of Columbia. In addition to that, I distributed surveys through a variety of electronic means and received 299 survey responses. This chart shows who completed the survey, from extension professionals, whether they work with growers or home gardeners, to commercial agricultural producers, gardeners and master gardener volunteers, people who work at schools or museums or parks, other places, and then an other category as well. You can see that the vast majority of survey participants were gardeners or master gardener volunteers. And what you can't see on this chart is that the vast majority of participants were from the state of Pennsylvania. So I really appreciate people in Pennsylvania who helped distribute this survey for me. On the survey, I asked questions like, where do you find information about biological control right now? And then, are you using this website currently? So almost half of the respondents are currently using either land-grant or extension websites to find information about biological control, which is great. But the vast majority of them have not used this biological control website, and not surprisingly, if they haven't used it themselves, they haven't recommended it to anyone else. The focus group feedback I collected was much more qualitative rather than quantitative. And this slide shows some of the themes that rose to the surface through multiple focus groups and were mentioned by multiple people. So during these focus groups, I invited participants to answer some questions for me, and then also try using the website and then give me some feedback on their experience. Overwhelmingly, focus group participants asked that we emphasize practical information, that they wanted to know, where do I purchase biological control agents? Exactly how do I use them? How do I apply them? Do they work? Are they compatible with fertilizers or pesticides? I was encouraged to think about the language I use and how language can either be approachable or it can push people away, especially if there's too much scientific jargon and I'm trying to attract audiences who are not necessarily scientists. Participants helped me come up with a list of biocontrol agents that they would like to learn more about that aren't currently on the website, and that includes especially biopesticides and biocontrol agents for greenhouses. I was encouraged to think about how to simplify the existing profiles for biocontrol agents so that, again, it makes it easier for people visiting the site to find that practical information. And the growers in particular said that the site absolutely has to be mobile compatible because they will most likely be using it on their phones out in the field when they found a pest problem. It was very important to participants that I include and use common names of insects and not list them only by their scientific name. And I was also encouraged to think about, what if someone visited the site and didn't know the name of the pest? There was, not surprisingly, an ask for less text and more pictures and videos, and to overall just make it easier for gardeners or growers to find answers quickly. In particular, to improve the options for searching and the functionality of the searching so that you could search by different parameters like the plant or the pest. So a big thank you to the people who participated in these focus groups and the surveys, as well as to the Northeast IPM Center and the USDA for funding this project.
Wonderful. Thank you, Mara. That was interesting. And our last presentation of the morning is David Owens, and he's going to be from the University of Delaware, and he's going to be talking about slug sampling in the mid-Atlantic, EPNs and temperature. And this was a project funded by the Northeast IPM Center.
Good morning. My name is David Owens, and today I'm going to discuss a project that myself and Sally Taylor are involved with the Northeast IPM Center's support, and also the support of the Delaware Soybean Board, to understand slug and natural enemy phenology in mid-Atlantic field crops. Slugs are a significant pest in the mid-Atlantic. They are favored by high residue, no-till, and cover crop fields. Most slug problems occur in Pennsylvania, Delaware, Maryland, the Shenandoah Valley area of Virginia, and also in Ohio and the hilly parts of Kentucky and even Tennessee. Not surprising, these areas have some of the highest cover crop adoption rates in the country. Now, the frustrating thing with slugs is that their activity is very weather dependent. Warm, dry weather shuts slugs down. Cool, wet weather, or even warm, wet weather shortly after planting is the most conducive to developing a slug problem. And you can see some of the feeding impact that slugs can have in this picture from NRCS of a soybean field that has just been completely decimated by slugs. Now, the other problem with slug management is that rescue treatments are extremely expensive. Metaldehyde and iron phosphate baits cost about $2 a pound and are applied at 10 to 40 pounds per acre. So we can make a rescue treatment in corn because the growing point stays underground as it pushes the first couple of leaves up. And so you can see the defoliation and its severity in time to make a treatment to save that plant and that stand. But with soybeans, we can't. So if slugs feed on the cotyledon or on the stem, knocking the cotyledon off, that will quickly kill the plant or set it back. And so soybeans cannot tolerate the feeding that corn can. So one of the questions that is of paramount importance to us as extension educators is, when is the best time to apply a bait? One of the first articles on slugs causing issues in soybeans in the United States was written by Ron Hammond. And in one of them, he writes that bait applications timed to slug egg hatching were the most efficacious, which begs the question, can we predict when slug eggs hatch? In this picture, we've got two neonate gray garden slugs, one on the cotyledon here, this soybean, and the other underneath the D in garden. But right next to that second slug, you can see a couple of slug eggs. So we started monitoring about 15 fields combined in Virginia and Delaware beginning in the fall, using temperature sensors and sifting through residue to look for what species were present and what life stages were present going into winter. We resumed weekly visits in these fields at the very beginning of March. And when we go to these fields, we look for slugs underneath of shingle shelter traps, and also by sifting through the crop residue in square meter sections. This project also gives us a chance to interact directly with the farmers and advise them what we're seeing and what the field's slug potential might be as we start thinking about planting. So far, our numbers have been a little bit lower than what both of us saw independently last year, in part because our winter was a little bit cooler. However, gray garden adults are present in some fields, and some fields that we're monitoring do have a worrisome population currently. We haven't yet seen any neonates, but it's only a matter of time. Last year in a pilot study in Delaware, we visited a field where gray garden neonates really started hatching out right around the 16th to the 23rd of April, and it occurred over a very sudden period of time. While we are visiting these fields, we're also collecting the slugs and holding them to look for entomopathogenic nematodes. Previous agricultural entomologist with University of Delaware, Ivan Hiltpold, conducted a survey in 2018 of 9 Delaware fields and showed that in several fields, up to 7 or slightly more than 7% of slugs were infected and quickly died from nematode infection. We do not know anything about these nematodes, and so we're using this project as an opportunity to learn a little bit more about their distribution and going forward, provide a baseline for nematode activity. If you'd like to know more about this project, feel free to contact either myself, Dr. David Owens, or Dr. Sally Taylor anytime. Thank you very much for your attention. I'll be happy to take questions.
Terrific. Thank you very much. So that was a really varied and interesting array of presentations. And I do not see any more questions that have come in. So if you do have any more questions, pop them into the Q&A feature. And but meanwhile, I can finish up some materials that I did want to share with you. So if you would like to create I'm going to share my screen actually, hold on. There we go. If you would like to, there we go. Okay, you should be able to see that now. If you would like to create a profile on our website for finding colleagues, it's a good way of connecting with people in the field. Find out, we encourage growers and researchers and extension people, all kinds of folks to put a profile up there. It's a good way of finding each other. There is going to be an email that will come out to you at the end of this session, asking you to fill out a short survey for us about the presentation, what we could do better, your reaction to the presentations and the format, and we'd love your feedback. Or feel free to email me with your feedback. I know some people for some reason had a problem logging in this morning. I have no idea why that was. Some people seemed to get in and others couldn't, so I apologize if you were one of those that struggled. The recording from this morning's webinar is going to be posted in a few days. We'll do a little bit of editing for it, so it won't be up immediately. And as soon as it's ready, I will send you an email with a link to the recording. And all of this work and many of the presenters actually today, at least a third of them, were funded through a grant that we received from USDA-NIFA. And without that grant, we wouldn't be able to make this possible. So I appreciate your participation, and I appreciate all the time and effort and thousands of hours of work that have gone into the presentations that we heard this morning. And it's fun to hear people take something that's taken hours, days, months, and years and distill it down into 5 minutes. And so I appreciate it. I know it's challenging. I'm a writer by trade, and it's so much harder to do something short than it is to do something long. So I appreciate everyone's participation. And with that, I will.
Jana, there is one question in the chat from Chang Lu to David, because their panelists can't use the Q&A. So the question is from Chang Lu for David Owens. Besides vegetation, what other factors affect abundance of slugs?
No-till. Fields that are in no-till production favor slugs. We do not see a whole lot of slug injury in fields that are tilled regularly, and high residue, and also fields with cover crops. And those last two go hand in hand with no-till production.
Okay, a follow-up. So does the presence of birds or mice reduce the abundance of slugs?
Our most important natural enemies are ground beetles, carabids, and Sally Taylor had a master's student show that fields that received an insecticide treatment with early cover crop burn down tended to have more serious slug injury than fields that did not. And it's a fairly common practice. Folks are concerned about having potential for cutworms or other pest defoliators in those cover crops. And it's a very cheap tank mix.
Okay, thanks.
Okay, thank you very much. Okay, well, with that, we will declare this complete. Thank you, Nancy, for pointing that out to me. And I hope you enjoy the rest of your afternoon, and we'll all soon be in the fields and out and about and enjoying the sunshine if you're not already where you are. So thanks for your participation. I look forward to seeing you again. We'll plan on doing this again in the fall, and slots for the presentations fill up really fast. But I will send out an invitation to everyone so that you can present again in the fall if you would like, or forward it to other people who you think would be interested in presenting. All right, and with that, I will say goodbye.